Okay. Well, let's go in and get started for today. So, I don't have the exams graded. Okay. So I'm about 2/3 of the way through that. It takes me about an hour and a half, 2 hours to get through one of those short answers. So I have meetings until about 4 this afternoon, so maybe I'll be able to get to that last one today, and then hopefully try and get those grades out to you tomorrow or Friday. Okay, so that's the plan. I think it's really quite optimistic to think that they would hit the grade book before probably late tomorrow or early Friday. Okay, all right. You do not have a quiz this week, so quizzes are usually due on Wednesday nights. We haven't obviously covered any new material, so no quiz this week, okay? You have a quiz due over this material on next Wednesday, okay? All right, so our riddle, what goes around the world but stays in a corner? A stamp. Exactly right, a stamp is the right response. Okay, so lecture 5, we are not leaving the topic of carbohydrate metabolism by any means. We are stepping into kind of a different aspect of it, and that is going to be a discussion on the regulation of glucose metabolism. So let me go ahead and start off by doing this, essentially. So let's think about here glycolysis. And I'm going to draw this out. And I want to address this because, like I said, I'm 2/3 of the way through grading the exam. And the second question on the short answers went well. The first question, not so much. Okay, so I want to go ahead and address that first question because it is certainly going to be something that even affects how we think about this. So if we are to go through glycolysis, glucose, right, goes to 2-pyruvate, okay, and 2-ATP are made in that process, as we have discussed in the glycolysis, and 2-NADH are made in that process as well. Okay, now, on the exam, I went on to say this, that the standard free energy change for this is about negative 135 kilojoules per mole. Okay? And that question is in regards to, okay, what's a steady state? And how does this exemplify a steady state? Considering that in the neuronal cell there's about 1 to 2 millimolar concentration of glucose. Okay, so let me ask you this question. You're saying, oh great, another question on a question. Precisely. Okay, so I'll give you about 2 minutes to answer this. I don't have a timer on it. I'll just time it on my watch. But 2 minutes, answer this question. So it says this. The standard free energy change is not going. There we go. The standard free energy change, what glycolysis indicates which of the following. What do you think? The reaction has a large KEQ and will go to completion or close to it. The reaction has a fast forward rate. If glucose concentrations in the cell are 1 to 2 millimolar, then the reaction is not at equilibrium. A, B, or C, or some combination of the above. Okay? All right, so go ahead and try and answer that question. Like I say, I'll give you about 2 minutes starting now. Okay? Is it locked? Is it still locked? Is it? Okay. I was wondering, no one's responding. Okay, there we go. Okay. You can talk to your neighbor about that as well, and what you put on the exam. Okay? Yeah, about a minute left. So, you're all right. Keep talking about it. Okay, you have 15 seconds left, so submit an answer if you haven't done so. And we'll check this out. Okay, 5 seconds left. All right, 4, 3, 2, 1. Okay, I'm locking it up. Okay, let's check these out. Let's check these results out. Okay, so 34% of you say that all of those are going to be true. 25% say that only A and C are true, and then only B and C true, 13%. So I'm going to agree with 25% of you on this one. Okay, so the standard free energy change does not give us any kinetic information. All it gives us is thermodynamic information. Okay, what it does tell us is, first of all, the reaction, let's go back to my slide here. Okay, and this is a really important point. This reaction, if allowed to reach equilibrium, will go to virtually completion, meaning that there will be hardly any glucose left. Okay, this delta G, standard free energy changes, tell us really the magnitude of the equilibrium constant. Okay, and negative 135 kilojoules per mole means that the magnitude of this equilibrium constant is really big, very, very large, which means that it will heavily favor pyruvate, NADH, and ATP, and there will be hardly anything left of glucose. Okay. So when we look at a cell then that has 1 to 2 millimolar glucose concentration with this reaction going on in it, what that means is that that cell, in regards to glycolysis, is not at equilibrium, it's rather at a steady state. A steady state is a non-equilibrium state of the system. Okay, and that's what that question was about, and that's what the data that I was trying to get you to recognize in that question is telling you. This tells you it should go to completion. We have data that suggests it's not at completion because there's 1 to 2 millimolar of glucose left in the cell, which means that this exemplifies a steady state. Okay, and you could have honestly answered that in maybe a sentence or two. Okay, now again, rate, the delta G has nothing to do with rate. That's a common misconception. Right? When you look at delta G values, it has nothing to do with how fast or how slow something will be. Okay, just straight delta G values. Okay, now the last part of that question then went on to say, well, how can a steady state like this be maintained? Considering that there are, in particular, that there are enzymes that could bring this to equilibrium very rapidly. And the answer to that is through regulation. And that's why I wanted to discuss this right now. Right? There are 3 enzymes in glycolysis. I asked you to name one of them, hexokinase, PFK1, or pyruvate kinase, that can be regulated, slowed down on purpose to maintain a fairly high level of glucose. Okay? And they can be regulated by those 5 mechanisms that we talked about: cooperativity, allostery, covalent modification, and genetic expression, changes in genetic expression. I said 5, there's 4. Okay. All right, so that's what I was essentially looking for on that question. When we look at like a neuronal cell or any cell, because they all have some fairly high level of concentration of glucose, what we see is that there's this constant, very large thermodynamic potential to produce pyruvate, which means there's always the ability to produce ATP through this reaction. Once it comes to equilibrium, there's no capacity using this reaction to produce ATP. And if you can't produce ATP in a cellular environment, the cell is dead. All right? So regulation, the topic of what we're going through in Lecture 5, is very important because it maintains these steady states, this capacity to make ATP. And it will make it at quicker or slower rates depending on what the cell needs, okay? Generally speaking. All right? Questions on that? Okay. Right off the bat, we wanted to deal with that because the answers were all over the map on that one. It was very difficult to grade. Yeah?
So on the exam, they have the true for question asking that, so you give the delta G is negative.
Uh-huh.
And then they're asking about the rate and the Hill coefficient. That's that. So how is linked to the coefficient?
Right.
The Hill coefficient.
Yeah, so we did talk a little bit about the fact that, so this is a standard free energy change. We talked a little bit about the fact that there are going to be actual free energy changes that are exergonic. Okay, those and I went on to say this, that the irreversible steps in the cell are catalyzed by enzymes that are slow. Okay, so just by looking at the delta G's, you couldn't say that kinetically they're slow. Okay, I mean, mathematically you can't get that, right? So when we though find an irreversible step in a cell, meaning that it only has one direction, the reason that it's irreversible is because the enzyme has been purposely slowed down. Okay, right. So.
But I had to tell the Hill coefficient though.
Okay, so I don't remember the exact question that you're talking about, but the Hill coefficient. So the Hill coefficient relate to the type of enzyme. So it's a cooperative enzyme. Okay, and Hill coefficients, if you have, again, not standard free energy, but actual free energy changes, those types of reactions are catalyzed by, or can be catalyzed by enzymes with Hill coefficients above 1. Okay, because that's a type of enzyme that can be regulated, can be slowed down. Okay. Okay. All right, now, so that sets us up now. Okay, and hopefully that's something that you can gravitate to and understand, because moving forward that will be very helpful as we start to look at mechanisms for regulating glycolysis and regulating gluconeogenesis, because it's going to become fairly complex fairly quickly. So we're going to go through in this lecture, first of all, I will just summarize, this is how glycolysis and this is how gluconeogenesis on a large scale are regulated, okay? And then we will move from that point and we'll look at specifically insulin and glucagon signaling. We'll talk about mechanisms that are used for insulin and glucagon signaling. And then we'll move from there and talk about tissue-specific regulation patterns, okay? And then from there we'll address some things, trying to apply some of these topics to like exercise and to diabetes, okay? All right, so let's do this, summary slide, okay? 2 summary slides. First of all, this is just a table. It's found in the Lineage's Principles of Biochemistry book. And I've already mentioned this, so I'm not going to spend a lot of time on this, but there are 3 steps in glycolysis that are sitting at irreversible conditions in the cell, meaning that they are catalyzed by enzymes that are indeed being regulated. And we can look at the delta G's actual free energy changes of these and see that they are indeed irreversible, and we can say that those then are the most important points of regulation in the pathway. Okay. I don't have a table like this for gluconeogenesis, but the opposing steps, the bypass steps in gluconeogenesis would be regulated as well. So what would be considered the pyruvate kinase bypass, or the PFK1 bypass, or the hexokinase bypass. Okay, this pyruvate kinase bypass catalyzed by pyruvate carboxylase and PepsiK, FBPase1 bypassing PFK1, and hexokinase is bypassed by glucose 6-phosphatase. Okay. Those are names of enzymes that you probably want to put in your mind, where they're going to come up time and again, okay, in upcoming discussions. Right? Now, if we are then to look at this is a summary slide and a kind of large picture view of how these 2 different pathways are regulated. Glycolysis on the left, gluconeogenesis on the right. Right? And you see by each of these enzymes, I don't have the names of the enzymes up, well, not the top one anyway, we have the names of this one, PFK1, right, and FBPase1 and pyruvate kinase, and the others down below, PepsiK and pyruvate carboxylase. All right, but you have all these other things listed by each of these reactions. Those things are things that regulate these steps, and you can see that there's actually a large number of them that regulate this pathway. It can be fairly complex. Okay, now, we are in this class not going to go through every aspect of the regulation of these 2 pathways for a number of reasons. We'll go through some of them, but not all of them. Okay, we're certainly going to mention some of them. Okay, but let me just start off with this first. You'll notice here that there are things that are listed by these steps in either red or green. I'm going to go ahead and underline all the ones in red, because some people have a very difficult time telling the difference between red and green. Okay, so I'm going to underline everything in red. Okay, all those words that I just underlined, or the ones, if you can differentiate green and red, are things that inhibit that particular step. Okay? And there's a lot of it. Everything that I didn't underline that's in green or has a positive or a plus sign next to it are things that activate that particular step. Okay? Now, using this summary slide, there's a couple of things that I want to go ahead and illustrate. So let me go ahead and first of all do this. Let me ask you another question. Okay, looking at that slide here. Okay, I'll go back to the slide here. I'm going to talk a little bit about this citrate. Okay, and let me just see how familiar you all are with the molecule citrate. What pathway is citrate produced in? Text me your answer and decide why this might impact glycolysis and gluconeogenesis flux. So just tell me what pathway it is, and just text me the answer to what pathway is it in. Talk to your neighbor about, well, why do you think that would impact glycolysis and gluconeogenesis? Okay, so I will give you, I don't know how long I'll give you, 2 minutes to do this. Okay, so text me an answer, what pathway is citrate produced in, and then just talk to your neighbor about why would that affect glycolysis and gluconeogenesis. Okay, all right. What do you think? We haven't talked about this pathway in this class, by the way. Okay, about 20 seconds left, so go ahead and text that answer to me, and we'll check this out. See what people are saying. All right, 5 seconds left. 2, 1, let's check it out. Okay. No clue, brain dead. I don't know why that one got highlighted. I didn't do that. It just, right? All right. That's the way it feels 5 weeks in, right? Yeah, you're like, okay, I need a spring break. It can't come fast enough. It looks like a lot of you are putting the answer that I was hoping that you would put. Citric acid cycle, TCA cycle, Krebs cycle. These are all the names of the same cycle, okay? As I look through this, yeah, I mean, that's the major answer, right? Okay. All right. Karen cycle. Well, that's an interesting one. I've never heard of that cycle. It's a new one. Or maybe we have someone that's just discovering a new metabolic pathway in their research lab, and they have discovered the citrate is produced in that pathway, and they're going to name it Karen cycle. If that is the case, let me know. We'll maybe try and section off a class and talk about Karen cycle. Try, okay? I can't promise anything, okay? But the Krebs cycle, citric acid cycle, that's right? So let me go ahead and do this. There's a reason that I wanted to bring that up here, okay? So this is something that we can do, okay? Here we've got glucose, okay? Glucose goes to 2-pyruvate. Drawn this a number of times. We've got ATP here that can be made, as we have noted, okay, by this. I will leave off the NADH, though it can be made as well, okay? And then what we will go on and talk about, again, high resolution structure of the mitochondria here, but what we will go on and talk about and see in this class is that pyruvate can be shipped into here. We've talked a little bit about that. Okay? This can be converted to something called acetyl-CoA. If you look back at that summary slide, you'll see acetyl-CoA plays a role in the regulation of some of those pathways. Right, but then acetyl-CoA is going to enter into, it's going to combine with oxaloacetate, and you think to yourself, that sounds strangely familiar. Yep, that's right. Oxaloacetate keeps coming up, and that's going to produce citrate. Okay? And then from citrate, we go essentially back to oxaloacetate. This is that cycle, that Krebs cycle. Okay? And I guess if we want to be stoichiometrically correct, we'd have to put 2 here. Okay? Now, one of the major outcomes of this pathway is NADH. Okay? And NADH, through processes that we have yet to talk about, will lead to ATP production. Okay? This pathway also produces a small amount of ATP directly. Okay? Now, what we are saying here, if you look at this, citrate, interestingly, will come up here and it will inhibit this pathway. Okay? At the same time, right, gluconeogenesis, the opposing pathway, that same molecule can activate that pathway. Inhibits this pathway, activates that pathway. Now, the reason I'm showing you this and the reason I wanted to talk to you about this is because this is one of many, 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 many examples that we could give of something that would be called feedback inhibition. Okay? The idea that citrate will come back and inhibit glycolysis, right, is really a direct result of the fact that if there's a significant amount of citrate in the cell, then that would be a communication from the cell to glycolysis that we have plenty of material to make ATP. Don't keep using glucose to make ATP. Store it or do something else with it. Okay? Citrate is also an indication in the liver cell that lipid biogenesis is increasing. Okay, so one of the things that would be important in that regard would be to slow the production of pyruvate down, right? And you, again, store glucose as, like, let's say glycogen or something like that in the liver. Okay. Of course, high citrate levels, okay, can also be an indication to, in particular in the liver cell, let's go ahead and take some of that pyruvate instead of making fat from it, because we've got plenty of resources to make fat. Let's make some more glucose out of it. Okay. All right, so that's the type of logic that we will see, and that's the type of thing that we will talk about in this, right? And this is just one example of this. Coming back to this slide here, I mentioned citrate, and you see these two things right here. Okay. And feedback, inhibition, feed-forward activation, these are common mechanisms. Okay. Now, that's one aspect as we look at a summary slide of glycolysis and gluconeogenesis, one aspect of regulation that we'll consider. And it's exemplified by citrate. We actually will not talk about citrate anymore. I mean, that's the last time we'll talk about it in terms of regulation of these pathways. Right? Now, as we look at other aspects of this pathway, again, just to summarize some of this, right? Citrate, as well as like in particular ATP and AMP, these are typically thought of as allosteric activators or inhibitors of these enzymes that they're next to. Okay. There are, you'll see here, and this is, we're going to spend quite a bit of time on this. I'll box them all in. Glucagon and insulin are listed in a number of different places. Okay. Presumably playing roles in either the activation or the inhibition of those steps. We talked about allosteric activation. Glucagon and insulin can lead to covalent modifications of these enzymes. For instance, we'll look at how glucagon can lead to the phosphorylation of the enzyme pyruvate kinase. Covalent modification. We can also talk about how glucagon and insulin will regulate genetic expression of the enzyme PepsiK. Okay. One of the main ways that the gluconeogenic pathway is regulated within a cell is through genetic expression, changes in genetic expression of PepsiK by glucagon and by insulin signaling. Okay. Those pathways lead to changes in that genetic expression. Okay. rather slow, but it can still do it, and it's fairly effective over long periods of time. Okay? Right. So those are the things, again, this is just a summary that we're going to be looking at. Allosteric activation, inhibition, through feedback, feed-forward activation type things. Okay? Covalent modification, changes in genetic expression. Again, those are topics that we talked about all the way back in lecture 2. Okay? Now here's where it becomes really fun. Okay? Just to mention one more thing that we're going to have to keep our eye on. Glucagon can lead to the covalent modification of pyruvate kinase in the liver, but not in any other tissue. So it becomes tissue specific as well. And you really are happy to be in the 21st century because we've discovered all this wonderful stuff, and it's become more and more complicated. 40 years ago, this would have been a different list. Okay? It wouldn't have taken quite as long to get through. Okay? So those are all things that we're going to have to keep in mind, and that's why some of what we're talking about is going to be essentially looking at tissue distribution and how different tissues regulate these pathways in different ways. Okay? And by the end of it, you may want to pull your hair out. Okay? All right. It's going to be fun. Promise. It's fun for me. Okay. Questions? Okay. That's the summary. That's where we're headed. Let's do this. Let's be more specific, and let's start talking about glucagon and insulin because of the importance of the role that they play in affecting glycolytic and gluconeogenic flux in a variety of tissues. Okay, not only that, but there also there's a very important disease state, diabetes, that we need to talk about in this class that's related to these signaling mechanisms. Okay, so let's do this. Insulin, first of all, I think I mentioned this before, but it's a peptide hormone, 51 amino acids long. Okay, and when excreted by the pancreas, it's excreted by the beta cell. Okay. In the pancreas, it can have multiple tissue targets: the liver, the muscle, adipose tissue, cardiac tissue, and a host of others. Okay, there's lots of tissues that have insulin receptors on them. Okay, and it will have pretty broad effects on those different tissue types. Okay. Glucagon here. Okay. This is also a peptide hormone, 29 amino acids, smaller than insulin. Okay, primarily affects liver tissue, adipose as well, but the liver tissue is really the main effect. It is excreted by the alpha cells, okay, of the pancreas. We won't talk about somatostatin in this class, okay, but that's also an important hormone regulator. So those are our two hormones that we'll spend quite a bit of time talking about in this class, moving not just into carbohydrate metabolism, but all the way into lipid metabolism, not so much in amino acid metabolism, but certainly in lipid metabolism. Okay? Right. Now, in terms of their overall effect here, and these are summary slides of what insulin and what glucagon can do. I certainly don't think I would want you to memorize this. We're going to cover some of these as we move forward. Okay, but they have really antagonistic effects on different tissue types. Okay, so let me go ahead and highlight this for you. For instance, let's just highlight this one. Okay, insulin. This table is about insulin. Okay, this table is about glucagon. Insulin will lead to glycogen synthesis in the liver and the muscle, whereas it will lead to the breakdown of glycogen in the liver. Okay, and that's typically how we think about these two. They have kind of opposing effects. Insulin is excreted in response to a fed state, high blood glucose levels. Glucagon is excreted by the pancreas in response to low blood glucose levels, usually in a fasted state. Okay. Now, as you start to look at insulin at the top there, you'll notice that, right, it's going to lead to glucose uptake in a variety of tissues. It'll lead to glycogen synthesis, okay? Certainly an increase in glycolysis and fatty acid synthesis, as well as triacylglycerol synthesis, okay? Those are things that, again, we'll talk about later on. Those are our target enzymes. If you think right now to yourself, insulin leads to primarily anabolic type processes, that would be a good way to think about this, okay? It's a sign or a signal to most tissues that now is the time to go through processes that will lead to growth and proliferation. Okay. In fact, in many, this is just how it affects metabolism, but in many cases, insulin can actually lead to growth and proliferation of cells. Okay. In some ways, can be thought of it as growth hormone. All right. Glucagon, because it doesn't affect as many tissues, really is saying, whoa, there's not very much energy around at this point in time. And in particular, the liver, because that's its primary tissue target, is just going to say, do whatever you can to ship glucose out to us, the rest of us. Okay, that's what the signal is. Okay, and you can look at that here. Glycogen synthesis goes down, glycolysis goes down, gluconeogenesis goes up in response to glucagon. It will also mobilize fatty acids in adipose tissue and something called ketogenesis, which we'll talk about later on. Okay, those are broad scale effects of these. Now here's the fun part. We want to look at how, right? And we're going to leave it at this, and we just say those arrows are nice, but how is it going to do that? What are the signals that lead to this? So we want to talk about insulin and glucagon signaling, okay? But before I do that, are there any questions on that? Nope. Okay, let's do this. Let's go to this slide. This is a decent slide, I think, that gives us a fairly decent introduction to insulin signaling, okay? The mechanism. If you've taken a cell biology class, you've maybe talked about this, okay? The biosignaling pathways. Biosignaling pathways are lots of fun, all right? So what I'm going to do is I'm going to cut this down the middle like this, okay? And we're going to look here first at insulin. Insulin will bind to the insulin receptor. INSR means insulin receptor. If you're super curious, the insulin receptor is a tyrosine kinase receptor, okay? And when insulin binds to that, right, step number 1, I have this written down over here. Insulin binds to the receptor. Step number 2, the receptor autophosphorylates itself. Number 2. Okay. Insulin receptor and tyrosine kinase receptors can phosphorylate themselves. Okay. Now, once phosphorylated, the tyrosine kinase receptor will do a variety of things. I put this big black line in this because everything to the left of that line is stuff that we're not going to really talk about in this class. Okay. It's important, no doubt, but we're not going to talk about it. Okay. The pathway of insulin signaling essentially bifurcates. It goes in 2 different directions. Okay. One of them, and this is what I was saying about growth and proliferation, one of them leads to the activation of the MAP kinase pathway. MAP kinase stands for mitogen activating protein kinase. Okay. Mitogen activating meaning mitosis activating. Right? Grow and divide, grow and divide. That's what that means. Okay. And that's a fun pathway because it has this MAP kinase that activates MAP kinase that activates MAP kinase. And then that does fun stuff. Okay. So you can thank me later for not going through that. You're welcome. Okay, now, let's go to what's on the right. There's a lot more on the right that we care about in terms of how it will affect metabolism. We're not going to talk about all of that on the right, but we're certainly going to talk about some of it. Okay, once phosphorylated, step number 3 is this. The IRS comes to the door. Okay? No, exactly right. IRS does not stand for Internal Revenue Service that's here to take your money. Okay? But it does stand for insulin receptor substrate. Okay, it's a protein, insulin receptor substrate. When the tyrosine kinase insulin receptor is autophosphorylated, insulin receptor substrate will be quote unquote recruited, meaning that it will stick to that. And then the tyrosine kinase receptor will phosphorylate it. Okay? And that's what you see here, these phosphates. IRS recruited receptor and phosphorylated. Now here's where it becomes real fun. We get some more abbreviations. Step number 4. Something called PI3 kinase is recruited to the membrane. Here's PI3 kinase. So this is step number 4. PI3 kinase recognizes phosphorylated IRS, comes to the membrane, if you will, recruited to the membrane. If you're wondering what PI3 kinase stands for, it stands for phosphatidylinositol 3 kinase. It's fun. Phosphatidylinositol 3 kinase. Okay? Phosphatidylinositol 3 kinase. Now, that's the enzyme. Now, here's what it's going to do. Okay? I'll start to run out of colors. PI3 kinase is going to phosphorylate PIP2, making PIP3. So let me say that again. PI3K phosphorylates PIP2, making PIP3. Don't you love biochemistry? I mean, there's not many places that you can say a sentence like that. I mean, let's be honest, okay? Right? So let me go ahead and highlight that. Here. That's number 4. We make PIP3. Now, what in the world are you talking about? What is PIP2 and what is PIP3? All right. Phosphatidylinositol 4,5-bisphosphate. This is PIP2. And you are looking at it right here. It is a phospholipid. It's found in the membrane. Okay? Phospholipids have, of course, they have the 2 tails. That's what you're looking at here. And then they'll have that glycerol backbone, and then there's a phosphate on the carbon. From that phosphate, there will be some group. In this case, it's the sugar inositol. Right? So that's this guy, inositol. Okay? That sugar in PIP2, phosphatidylinositol 4,5-bisphosphate, has a phosphate on the 4th carbon of the sugar and the 5th carbon of the sugar. 4 or 5. PIP2. PIP3 is simply the addition of a phosphate right here. And I'm going to have to draw it over here, but we'll just draw a P with a circle around it. That means phosphate group. Once I added that, this becomes now PIP3. PIP3 is made by PI3K. And we call this then PIP3 phosphatidylinositol 3,4,5 triphosphate. Okay. And so that will start to be enriched in the membrane. So, summary. Insulin binds, receptor autophosphorylates, IRS is recruited to the membrane. It phosphorylates, that recruits PI3 kinase. PI3 kinase starts to make PIP3. Right? So the membrane starts to have a bunch of this inositol sitting outside of it that has this 3rd phosphate on it. Okay? That then, we're not done. No, we're not done. Sorry. Step number 6, PIP3 is a substrate, if you will, or it's something that these 2 enzymes will bind to, PDK1 and AKT. So these are recruited to the membrane. Number 6. If you're wondering, wait, I got screwed up here. This should be number 5, sorry. Number 5, there we go. If you're wondering what PDK stands for and AKT stands for, PDK stands for phosphatidyl inositol dependent kinase. AKT stands for nothing that you really want to know because it's not helpful at all. Okay, when you know it. I can't even remember what it is myself. So somebody is saying, no, it probably is helpful, you just don't remember it. No, it's not. Okay, just trust me in this. Okay, it's not. It was named for some strange, it was not, the discovery of it led to some strange nomenclature for it. So, but anyway, PDK1 and AKT they will recognize accumulation of PIP3 in the membrane, okay? And they will come to the membrane and be active, okay? Well, in particular, PDK1 will be active. Now, here's step number 7. You say, oh, are we almost done? Kind of, okay? Step number 7 is this, and this. AKT will be phosphorylated, okay? So AKT will be phosphorylated by PDK1 and mTOR. Unfortunately, there's 2 versions of mTOR. Okay, so this happens to be mTOR-C2. Now, once AKT is phosphorylated, it does stuff. And we're going to leave it at that. Okay, because AKT then will go out into the cell and there'll be lots of targets for AKT and what it's going to do. Okay. Once it's phosphorylated, and then there's different phosphorylation sites on AKT, one that will be phosphorylated by PDK1 and one phosphorylated by mTOR. Now, when I say stuff, let me show you. Here's AKT. So this is the signaling pathway that we talk about. That's AKT, and then everything that has got arrows drawn from it, that's what it does. You can look at that all on your own if you want. We'll talk a little bit about what it does, okay, but the details become nauseating, okay, to say the least. It just becomes a whole bunch of boxes with 3-letter or 4-letter acronyms in them. And that causes you to say 4-letter words directed at someone like me that's going to hold you accountable for them, right? Okay, so we're not going to go beyond that. I mean, we'll talk a little bit about the end results all the way at the end of this slide, but we won't talk a lot about the stuff in between, okay? Right. So this is insulin signaling. What we just did essentially is this, okay? Now, I will say this, downstream effects, and come back to this. Survival, that's a good one, right? I like that one. We're not going to talk too much about that. But like, for instance, glycogen synthesis, glucose transport, right? The movement of GLUT4 to the membrane, okay? We'll talk a little bit about, it doesn't have on here, but glycolysis rate will be affected by this as well. There's other things that will happen. So we're going to look at some of those downstream effects that AKT will cause, okay? And I'm sure that this is going to intersect or has already intersected with some of your other classes, okay? These pathways, I mean, this is a pretty well-established pathway. And you certainly talk about it. If you're taking 465, if you haven't talked about this yet, you will. Okay, and some of the other stuff up here. Questions? Cell signaling. You gotta love cell signaling. Now, let me ask you this. Okay, I got a question for you. Karen's cycle, that's right. That's a great one, huh? All right. Okay, let me ask you this. So P10, I'm going to kind of summarize what we just did. P10 is a phosphatase that removes the phosphate from the 3' hydroxyl on PIP3. If P10 were overexpressed, meaning there's more of it than you'd expect in a skeletal muscle cell, what would happen to this cell's ability to absorb glucose when insulin signaling was present? Would absorption go up or would it go down in that cell? So insulin's there, but there's a lot of P10 there as well, and P10 is active, is more than you'd expect of P10. What's going to happen? And glucose absorption going to go up or down? I'll give you 2 minutes. Talk to your neighbor about that, okay? And we'll move on from there. And your marks get set, go. Just tell me, up or down? Up or down? What do you think? Okay, about 30 seconds left, so haven't quite submitted something, do so. Okay, 10 seconds left, so submit something quick if you haven't done so yet. 4, 3, 2, 1. All right. Let's check it out. Okay. Oh. Absorption. Go. Let's look at this. Okay, so up. Some of you say down, 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 down. Lots of downs. Absorption will go down, down, lots of downs. Some say up. Mostly down. Stay stilled. Okay. All right. Go down, go down. Okay, down, down. Okay, so most people are saying down, it would look like, just as a kind of a quick glance of all these things. Okay? So, yes. Yes. Both true and false at the same time. Some of you are thinking that's what I put on the exam. A and B. You can't say I'm wrong. Okay. All right. So I'm going to agree with you, the majority of you, not with yes. I'm going to agree with down. That's what I'm going to agree with. So let me go back here and show you this. P10, okay. P10, first of all, P10 is something that we find. It's a phosphatase. It's there in healthy cells. It's good. Okay, it's a good thing to have. It's one of the ways that insulin signaling is reversible. And reversibility of these pathways is very important. Okay, if you couldn't reverse this pathway, then we'd have sustained effects of these outcomes, and that would be very unhealthy for the cell. It would lead to a disruption of steady states and homeostasis and eventually the death of the cell. So all of these things need to be reversible. P10 is one of the mechanisms that this is reversible. It's an unregulated phosphatase, meaning that it's constantly active, okay? And so what it does is there's this constant, if you have insulin signaling going on, you're making PIP3, but some of it's being broken down by P10. But when insulin's present, if PIP3 is being produced at a faster rate than it can be broken down. Okay. Now, the problem would come if P10, this guy right here, were to increase in expression, meaning there's just more of it around, more than it should be around. If that were the case, then PIP3 would not accumulate or could not accumulate. And in skeletal muscle, that would lead to a lack of the movement of GLUT4 to the membrane. Okay? That skeletal muscle would be considered insulin resistant, which is the hallmark feature of type 2 diabetes. And there has been reports that have demonstrated that obesity can lead to the activation of an inflammation pathway that will lead to increased expression of P10. Okay? So that's one of the pieces of the puzzle of how potentially cells can become insulin resistant, is that they start to overexpress P10. Now, I will say this, type 2 diabetes is a very complex disease disorder, okay? And so we'll investigate some aspects of it, but P10 is an aspect of it, okay? There has been some that have considered P10 an important then drug target, right, for the treatment of type 2 diabetes and also some cancer cells, okay? Some forms of cancer. But we'll stop there, okay? And we'll step into, on Friday, we'll talk about glucagon signaling, the antagonist of this, if you will, okay? All right, have a good rest of your day.
That test question you were talking about
Yeah.
at the beginning. That was the whole point. Well, I mean, I get where you're going with it, but that part.